Hello, everyone. I hope you all had a great morning and that your day is going well. Welcome to the Global Plant Council webinar on Memories of Pathogen Encounters, Defense Priming and Epigenetic Regulation. So my name is Susanna. I am a publishing specialist with the journal Frontiers in Plant Science. This is the first webinar for our journal, and we are delighted to work with the Global Plant Council to highlight exciting new research and to support the plant science community. I am very happy to have you all here for what we believe will be a very fun and interesting webinar. So thank you so much for joining. And let me briefly go over our agenda for today. So we will first have a short introduction to our journal by my lovely colleague, Darina. And this will be followed by a short talk by Dr. Isabel Mendoza, the Chief Communications Officer at the Global Plant Council. We will then have our key speakers give their talks. And our key speakers are Dr. Nicolas Cecchini and Dr. Ho-Wan Jung, who are the winners of the 2021 Global Plant Council Prize, and who are kind enough to share their research in this webinar. They will be presenting their winning research topic. Nico will talk about the role of MOM1 on priming and systemic resistance, and Ho-Wan will talk about the role of HAC1 and DDL in plant immunity. After this, we will have a Q&A session where you are welcome to ask Nico and Ho questions, and we'll have maybe a little bit of an open discussion. So before I turn the floor over to my colleague, Darina, I'd just like to mention that this webinar is being recorded and that the video will be posted on our Frontiers YouTube page in case you want to rewatch the presentation. So please make sure that your microphone is muted and that your camera is off. You are welcome to submit your questions to the Q&A box at the bottom of the window during the talk. Just please specify who your question is for Nico or Ho-Wan. And our speakers will be happy to answer your questions at the end of the presentation. And yeah, if we have some time after the Q&A, we can also have maybe a little bit of an open discussion if our attendees want to do so. So now I'm going to give the speaker rights over to Darina, who will tell you a few words about our journal.
Do you know if Darina is on the call?
Hello, can you hear me?
Yes.
Can you hear me now? Yeah. Okay. Apologies for the tech issue there. So I'm Darina. Thank you very much, Susanna, for the short introduction. I'm a journal specialist at Frontiers in Plant Science, and I'd like to give a short introduction to Frontiers and our journal. So Frontiers is an open access publisher, and we have one goal: to make science open. So why is this our mission? Our goal is to live healthy lives on a healthy planet. And as research evolves and the scientific community develops new goals and strategies to tackle the many issues we face today, we want to make sure that everybody has access to this knowledge and can read and share it at any time. And how are we planning to do this? We are on the way to becoming the largest and most cited publisher in the world. We are the 6th largest and 3rd most cited publisher currently. Our articles have received 1.9 billion total views and downloads, which just goes further to highlight the power of open access. And to date, we have around 163 journals and are constantly developing by launching new journals and sections to home our groundbreaking research. And we now have a short video to give you an insight into frontiers in plant science. So Frontiers in Plant Science is the most cited journal in its field, and the recent impact factor recently released celebrate our new impact factor of 6.6 and site score of 8. Next slide, please. We publish research to improve our understanding of plant science, and to date, we have 24 sections that cover all areas of plant science. Articles submitted to the journal pass through a rigorous peer review process, and we also have an editorial board of experts who work alongside us to ensure that we publish the highest quality of research. And our research topics, also called article collections, are hosted by experienced professionals within the plant science field who work as guest editors to shape the scope of their collection and give a platform to other experts in the community to submit their research. Articles submitted to research topic have more views, downloads, and citations, and we give full control to our guest editors to shape their collection and handle the latest research papers. And there is also a chance to win the Global Plant Council Prize. Last year, this was awarded to Nicolas Cecchini and Jung, who edited their research topic on systemic resistance and defense priming against pathogens. There is still a chance to apply and win the Global Plant Council Prize of 2022 and share your research on our high visibility open access platform. So I hope that gives you an insight into frontiers in our journal. And I'll hand you back to Susanna.
Okay, thank you, Darina. That was a wonderful introduction to the journal. And now let me introduce our first speaker, Dr. Isabel Mendoza, who is the Chief Communications Officer at the Global Plant Council. We're so happy to have Isabel here with us. She will share with us about the Global Plant Council, their mission and their goals, and the activities that they organize for the plant science community. So Isabel, please feel free to start.
Thank you, Susanna. Do you hear me well?
Yes, yes, we can hear.
Yeah, thank you, Susanna, Mila, Lewis, Darina, and all the Frontiers and Plant Science team for organizing this webinar. My name is, as you already mentioned, Isabel Mendoza, and I'm the Chief Communications Officer for the Global Plant Council. Can we move to the next slide, please? And the first question would be, what is the Global Plant Council? So in case you don't know, the Global Plant Council is a global coalition of plant science organizations. Next slide, please. Currently, the Global Plant Council includes 27 organizations around the world. The members range from professional plant science societies, such as the Japanese, the Canadian, the Argentinian, the Chilean, the Australian, the Portuguese, and some more, to purely research organizations such as the Max Planck Institute for Molecular Plant Physiology in Germany and the Australian National University, and really anything in between that it's related to plant science. Next slide, please. So the top objectives of the coalition are to raise awareness about the importance of plant science for society and to build a sense of community for the plant science across the world. Our aim is to be recognized as a voice of reference for plant science and plant scientists at a global level. To achieve all this, which is quite major, we organize in-person activities at high-level conferences for the community, that can range from anything from a workshop, a panel, or a session. And we keep a strong online presence, including on social media. We are present in all major mainstream social media platforms currently. So I invite you to follow us in any of those. Currently, we hold something almost 35,000 followers, if I remember correctly. Next slide, please. So just to give a sneak peek of the activities that we carried out, here are some examples. One such is the Early Career Research International Network, or ACRI, addressed specifically to early career researchers to help them find a job, land a grant, build a network of collaborations, and communicate their science efficiently. The Frontiers Research Topic Prize by the Global Plan Council is framed under this umbrella, but there are many other activities that you can find easily by going to our website, specifically under Early Career Research International Network. Secondly, we also put in motion working groups to discuss matters of joint interest for the plant science community, bringing together experts from around the world to discuss the issues and organize events or put suggestions for policies in writing, whatever appropriate. Currently, we have one working group ongoing on digital sequence information, access and benefit sharing, and the Nagoya Protocol, and how this all these issues could impact the future of the work of plant scientists. Next, we also have a series of webinars that maybe you are already acquainted with, around mainly 3 pillars. One is the global collaborations, where we highlight transnational projects and how they came to be, the inception of the idea behind these projects, how they are managed, and how they are kept going, where do they fund the money, and how these relationships that started the global collaborations started, in order to inspire young researchers and also more senior researchers to start such big projects. So secondly, we have the Academic Odyssey series, where we invite outstanding researchers to present their career, how they started and how is it going. But we only invite people with an unusual career path, in order to also inspire researchers, young, in this case, young researchers, that there is not only one possible career path, that it's PhD, one postdoc, and then tenure, and that's it. There are many possibilities, and we want to highlight that. And thirdly, we also have a series on soft and transversal skills, anything that could be how to write papers, how to choose the appropriate journal to publish, how to review a journal, how to review a paper, and science communication in broad sense, anything that we think it could be of interest as a soft skill for the plant science community. And finally, as already more or less mentioned, when appropriate, we put in writing the thoughts of the community in the form of either a policy or a position paper, and then make those position papers known. For example, last year, in time for the COP meeting in Glasgow, we issued a climate change statement with the views of the plant science community, in the hope to influence the policymakers there. So next slide, please. So that's all from my side. If you want to connect with me or with the Global Plant Council, you may do so by using the different channels available. Here in the slide, you can see my email address, our URL. As I already mentioned, we are in most of the mainstream social media platforms, as you can see here highlighted: YouTube, LinkedIn, Facebook, Instagram, or Twitter, but we are also present in Weibo for the Chinese audience. And we are very happy to connect. So drop us a line whenever you think so. And thank you again to the organizers for the opportunity to present the Global Plant Council and what we do. And I think we are ready to hear Nicolas, Cecchini, and Jung talk about the memories of plant or memories of pathogen encounters.
Okay, thank you so much, Isabel, for sharing with us about the Global Plant Council. It's definitely a great resource for plant scientists. And so now, without further ado, I would like to introduce our first key speaker, Dr. Nicolas Cecchini. So Nico is an adjunct professor and associate scientist at the Konitset Center for Research in Biological Chemistry at the National University of Cordoba in Argentina. So I will now pass the presenter rights to Nico and let him share his presentation. Thanks a lot, Nico. And yeah, please feel free to share your screen and continue.
Okay, let me see. Can you see my screen? Hello?
Yeah, we can see the screen.
Okay. 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 Hello, everybody. First of all, I want to thank the Global Plant Council and Continuing Plant Science for this very nice webinar and to have the opportunity to present some of the results from our lab. So today I'm going to talk about MOM1, that is a chromatin remodeling factor, a protein, and the possible role that this remodeling factor could have in the induction of priming against pathogens and particularly in systemic resistance of plants. So as a quick introduction, before I start with the results, I want to present the plant immune system. As you may know, or most of you, the plant immune system can be divided into parts, like the constitutive defense in one side, that like the cuticle or the cell walls that actually stop most of the pathogens. You've never seen like a lot of plants with infections. But then we have the other part of the immune system, that is the inducible defenses. And this part rely basically in immune receptors. So these sensors that can recognize the pathogens. In plants, we can find two kinds of receptors, the PRRs, or pattern recognition receptors, and the NLRs that are intracellular receptors. So just to show you more really quick how it works, the recognition, I can show you this cartoon from Petersen that I really like. You can see here in the plasma membrane the PRR receptors. These receptors recognize usually conserved molecules in the pathogens that are called PAMPs or MAMPs. And as you can see here, the PRR is recognizing the flagellum from a bacteria. So this receptor trigger inside the cell like defense signaling that end up with the immune response. Of course, in the evolution, some pathogens learned how to downregulate this first layer of defenses, and actually they do this usually injecting or secreting virulence factors or effectors, as we call it in the plant field. And actually, these effectors basically they downregulate the immune response. No, oh sorry. But of course, there is another layer, as I told you, in plants that are the NLRs. These are really specific receptors, and they recognize some of these effectors that are in some strains of the pathogens, and they trigger a really strong response that usually is associated with cell death in the site of the infection. But actually, the immune system in plants is more complex. And actually, after the recognition of the pathogen or even a microbe in leaves, on the roots also, most of the plants trigger, sorry, I'm having, I don't know what, okay. So after the recognition, plants trigger the production of many molecules, and some of them are mobile signals that go through the plants to the systemic tissue, and they induce this systemic resistance. This systemic resistance, most of the time, is associated with the priming or the immunological memory. That is the topic of my talk today. So what is this priming state means? Basically, that when a secondary infection arrives or a pathogen attack for a second time, the plant, because it remembers the first encounter with the pathogen, it triggers a defense that is more efficient. Basically, usually it's faster and also higher. No? So the plant became more resistant to the infection. So what are these mobile signals or molecules that are produced after defense? Well, we call it priming inducers, basically because if you treat the plant with this molecule from outside, even in the aerial part or in the roots, like watering, you can induce a priming or a prime state that is very similar to what you see in natural infections, in the natural systemic resistant induction. Two of these molecules that are also mobile signals are acylic acid or ASA and pipicolic acid or PIP that I'm going to talk during the talk, and other molecule that is not a mobile signal yet, it's in the future, it's aminobutyric acid or BABA, that is a kind of famous molecules for inducing priming, because not only induce priming against pathogens, but also against abiotic stresses. So why is important priming? No? Well, basically because to establish priming, you use really a little bit of energy. So it has a low energy cost. Why is this? Because when you want the plant that is in a prime state, actually it doesn't have the constitutive defenses. So you don't have all the defense genes in use, for example. And actually, if you analyze a plant with priming, you don't see many changes, at least transcriptionally. No? So this means that the plant is prepared for a new infection, but it's not wasting energy to maintain that. No? So if this plant that is prime is in an environment that there is a lot of abiotic stresses, it's highly probably that it will be infected again. So in this moment, the plant that is prime will have a much better fitness than a plant is not prime. So that's why it's so important. And actually, there is now a lot of effort in the world trying to find the best molecules that induce the best priming for a longer time. No, a chemical or some bacteria that can induce also the priming. So what are the changes that establish this priming, this memory? So basically, there are several reports, some of them talking about, for example, the increase of signaling factors, the latent signaling factor like MAP kinesis, for example, or increasing immune receptors. No, this make the plant even just to spend energy in the first moment, but then you have more receptors and more amplifying factors of the defense signaling. So you have a more efficient defense response. But of course, as you can imagine, most of the reports talk about the correlation between priming induction and epigenetic alterations of the chromatin. You can imagine you open the chromatin, or the chromatin, for example, the promoters of defense genes is more open. So then in the secondary infection, the gene can be induced faster or even higher. And actually, there are many reports in the literature about genetic marks changes that usually they correlate with the priming, more priming or less priming. And these changes, most of them are changes in methylation of DNA, in the cytosines, that you know, we can open or close the chromatin, and the modification in histones, particularly modification in histones. But even though there is a lot of reports about this correlation, actually there are almost no works where they analyze epigenetic factors or chromatin remodeling factors or complexes that are more specifically implicated in priming. What I mean with this is, for example, a mutant in a factor that affects the priming, or some factor that change the amount in the priming induction or so on. So one of these sorry, what is talking? Okay, one of these factors that was published in the plant group in my institute not long ago is Morpheus mollicus 1, that is actually a chromatin remodeling protein. From now, I will say just MOM1. And what is MOM1? Basically, as you can see here, is a protein with a lot of domains. It's a big protein, and it has at least 3 domains that are highly similar to other chromatin remodeling factor from the family, but also has some domains like this in blue that are unique for MOM1. No, I'm going to talk about that in a moment. So what's the main function described till now for MOM1? Basically is regulate the deposition of methyl groups in the lysine 9 of histone 3, no? And particularly in region of the genome that you have a lot of methylations of the cytosine. And those places usually are centromeres and pericentromeric regions. So basically the function of MOM is the repression of all the transposons that are in the centromere and centromeric, pericentromeric regions, as for example, the transposon TSI. Also is implicated in regulating other transposons in the genome, and also when a plant that is a transgenic plant, to regulate the silent genes. Sorry, somebody talk. Okay. So to show you the function, like in an experiment of MOM1, you can see here, for example, in this mutant in MOM1 that is called MOM1-1 that has an insertion in the C terminal, you can see in this RT-PCR how highly expressed TCI, this pericentromeric transposon. It's really from 0 because the centromeric transposon has to be completely repressed in Arabidopsis at least. And in MOM1, the transposon expressed a lot. Another very interesting finding about MOM1 and that it will be important for the talk is that several years ago, in the laboratory from Dr. Paskowski, that they did a lot of work with this protein, they found out that only the one domain is required to completely complement this repression of the transposons in the centromere. It's really quite interesting because you can ask what are all the other domains do, no? But anyway, this construct that is called minimum can complement completely the phenotype of the high expression of TCI in MOM1. So why I'm saying that MOM1 it could be negative regulator of priming in Arabidopsis? Well, first of all, basically, if you infect a MOM1 plant, a mutant plant in this factor, with a bacteria in this case, you can see that the plant is much more resistant. No, so it became more resistant. But this doesn't mean that it's priming because it could be constitutive defenses. No, what it's saying more closely that it's priming is that in the basal state, in optimal growth condition, in sterility, in this case, PR1 is a classic marker of defenses. You can see that in the basal state it's not induced. But when you infect after the infection of the plant, 8 hours post-infection, you can see a fast induction that is not occurring in wild-type plants. This is telling you that it is possible that the MOM1 is regulating prime. No? Most important is the fact that minimum, the plant that express this construct, it's a small construct in the background of MOM1-1, also complement the resistant phenotypes that we found in MOM1. No? Why MOM1 mutants are prime? We don't know yet. There are several hypotheses, and I'm not going to talk about that. If somebody wants to talk, we can do it after the talks, and we can discuss. But basically, one of the ideas is that MOM1 in non-sterile conditions have a lot of immune receptor expressed, highly expressed, and this is because there is a transactivation of this gene that is mediated by the expression of these transposons in the centromere. So what's our question? Basically, if MOM1 is really a negative regulator of priming against pathogen. With this, what I mean is, so it's implicated in the induction of priming with, for example, with priming inducers. like some classical ones like azelaic acid, piperacolic acid, and BABA that I showed you before. So what we did, we tried to study the immunological memory and priming that is induced or not in the mutants of MOM1 and in mini-MOM, no, with different treatments. And that's what are the results that I'm going to show now. Oh, I know what's going on. Okay. So the first thing that we did was try to check with an easy test if the priming of MOM1 and mini-MOM is changed in some way after the induction with this primer, with these inducers, no? So luckily for us, there was described some years ago that both ASA and BABA can regulate the growth of the principal roots of Arabidopsis on plates when you treat the disease in this inducer on plates. And actually this directly correlate with the capacity of inducing priming. What I mean with that is that mutant factors of the azelaic acid or BABA pathway, when they are mutated, the treatment on plates doesn't work, no? So what we did first, we set up the plate. Oh, sorry, we'll do it with my mouse. So what we did first was to, okay, it's something else that's going on. I don't know why. Okay, so we set up, sorry, I will stop this. I don't know what's happening. Wait a minute. Oh, sorry. I will restart the PowerPoint because something's going on. Okay, can you see the presentation?
Yes, we can see the presentation.
I think I'm having the same problem. Oh, I don't know why. Okay. Sorry about this. I don't know why this is happening, actually.
Nico, is it just that the rest of the presentation won't load?
Yeah, it is loading the slides automatically. I don't know why.
Okay.
Okay. I think I fixed it. Sorry. Okay.
No.
So, okay, let's start again. So what we did basically was to set up this plate assay for our mutants. So to do that, first we used the best growth condition to avoid the induction of basal defenses in MOM1-1. I told you that this mutant, when it's in soil, particularly, it induced by itself the defenses. So we use the plates, and as you can see, we check two genes that are the PR1 and ICS1 that are marker genes for defenses. And as you can see, MOM1-1 has the same level of defenses as the Zurich, that is what the wild type in this background. Also, mini-MOM doesn't show any defense inductions. So also we check some immune receptors that are possible targets on one, like RLK7, ADR1, and ADR1, and MOM1 are kind of important in receptors, and they're also not induced in the mutants. And also, finally, we check, of course, if TCI is induced in the MOM1, just to check that we are working with the mutants. No? So once we have the setup, basically what we did, we grow the seedlings in plates with different concentration of the inducer ASA and BABA. And as you can see here in these representative images, you can see first that in wild-type plants, both inducer BABA and ASA are working. The roots are shorter, but what is more interesting here is that actually MOM1, the mutant, it's more sensitive or is hypersensitive to both inducer, particularly to ASA. This could be kind of expected because we are saying that actually it's a negative regulator of priming. But it was not expected was that minimum this control is just one domain and complement all the phenotypes. Now, it's not only complementing this inhibition phenotype of the root, but actually it's not responding at all to the inducer because it looks exactly as the wild type. You can see this much better here in the quantification of the root length at different BABA concentrations and ASA. And again, you can see that in mutant MOM1, the mutant is much more susceptible to both ASA and BABA, and minimum, it doesn't respond at all. So our next step, we wanted also to check if this could be happening with pipecolic acid. That is another really important inducer of priming and actually systemic resistance also. So, but the problem with PIP is that there is no a previous report saying that this assay could work. Basically, there wasn't anything first if PIP also inhibit the root growth, and also if some of the factor in the signaling of PIP for mediating the priming induction is affected in this assay. So that's what we did here. And as you can see, first of all, the wild type plants is being affected by PIP, reducing the growth. But most important, FMO1, that is this factor that I'm telling you, it doesn't respond at all to the treatment on plates with PIP. No? So also PIP can be tested in plate assays. It's new, and we think that it's important actually. So basically, once we have this, what we did was the same assay, and again, it looks we have to do higher concentration now, but basically MOM1 is more susceptible to PIP treatment, and minimum it looks again that it's not responding at all. So okay, this all is good, but this is kind of indirect assay to just easy rapid and easy test the priming. No? So okay, what's going on with the real thing that is the systemic priming? So what we did, basically we water adult plants growing in the soil, normal plants with ASA, PIP, and BABA, and after 1 day we infect the aerial tissue, the systemic tissue, with the bacteria, with Pseudomonas in this case, and we check if the bacteria grow more or less. So if this prime should be more resistant to the growth. So first we did this with the mutant MOM1. As you can see in the wild type background storage, the treatment with the 3 inducers induce the resistance to the bacteria, as you can see in the gray bars. But when we did the same with MOM1, what we first observed, and I showed that before, MOM1 is more resistant, but the treatment with inducers don't induce more resistance than MOM1 had from the beginning. So what does it mean is that you can't prime a prime plant, no? So there is a limited, particularly we believe that when plants are growing in soil, because in plates we can see like a top effect on the root growth. So, but most important and what we want to check if it is minimum, it's really not responding to the inducer. I mean, what could mean this is that minimum is like a dominant version of MOM, or let's say it could be like a overexpressor, no? It's having like the super negative regulation of priming. So as you can see, that's what we obtain. You can see again that the wild type induces priming with the 3 inducer, but minimum again it's a plant that can't be primed. Actually, at least with these three inducers, which is kind of surprising and really interesting. Another way to see this is to calculate the response gain of priming. So it's like a number that tells you how much priming prime the plant is, and you can see that with wild-type plants in gray, we have a very good priming, but with in minimum, the priming is almost zero, particularly with acylic acid, and just a little bit with BABA and PIP. So we can say that minimum plants or minimum construct is acting like a dominant version of MOM. So finally, we wanted to check, okay, all this is really nice. MOM1 that was proposed as a negative regulator priming, is priming from the beginning, and minimum that is like a dominant version is not responding to priming. But what happened in a wild-type plant with MOM1? If MOM1 is a negative regulator, you expect that when you induce the priming, MOM1 activity will be downregulated. No? And the activity could be enzymatic activity, could be interaction with another factors, the nuclear localization changes, could be a lot of things, but it could be also that the transcript levels change. No, that's the more easy way to check something than the others. No, so that's what we did. Basically, we again watered the plants with the three inducers and checked the transcript level of MOM. And happily for us, we saw like a nice reduction of the transcript of MOM1 up one day after the treatment with the three inducers, as you can see here in this qPCR, around 50%. But it's a good reduction of the transcript. No, and what it was even more interesting and correlate with this is that in the same sample we can see like an induction of TSI, this pericentromeric transposon that is regulated by MOM. And what is even more interesting is that one immune receptor, RK7, is also induced when we treat with inducer. So we have a correlation, and really looks like MOM is like a central component regulating the priming by regulating the immune receptors levels. And remember, this is in the systemic tissue. We are watching the roots. So just to finish, we wanted to check if this induction of these immune receptors that are target of MOM are also being induced in a natural priming condition or state. And basically, this occurs in systemic resistant responses. For example, the systemic acquired resistance or SAR. No. So what we did, we looked for transcriptomes that they were they had data sets of systemic tissues of plants with SAR or priming induction, no, or systemic resistance. Basically, this experiment they infect a local leaf with Pseudomonas in this case, and then we check what's going on in the systemic tissue, no, in this RNA sex analysis. We found three studies where they analyzed this at least, and as you can see, again, RLK7 is induced in the three studies, in the three data sets in the systemic tissue, which it's very interesting because actually these experiments were done at 3 days after induction of systemic resistance. So it's very interesting that still at that point the genes are still induced, and the same happened with two others, NLR type immune receptors that are very important. So we really believe that actually MOM1 could be a negative regulator of priming for many types of priming induction. So our working model right now, that is more or less what I told you, is basically that during the onset of the priming induction, we have like a reduction of MOM1 levels. This triggered the induction of NLRs. All this is happening systemically. And the reduction of MOM1 also happened with the transient induction of the pericentromeric transposons. So I think we can say that actually, we can strongly suggest, I won't say indicate, that MOM1 negatively regulates the priming, at least triggered by ASA, PIP, and BABA, and in Arabidopsis. And with that, I want to thanks to all the people that was working in this project, and particularly to Julian Miranda, that he was an undergrad that started with this project. Now he's a grad student doing ecology, and he looks very happy. And of course, the amazing postdoc in my lab, Micaela Pepino, that she's doing a lot of work right now, and also Ignacio Lescano, that he's a postdoc in Malena Alvarez's lab, but actually now he's a researcher. Malena and Ignacio, they were working a lot also in this project. And finally, the amazing help from Damian Cambiano, that is a collaborator in the lab, that he was doing all the RNA-seq reanalysis of all those data sets. So with that, I finish and I open to question. And okay, I forgot. Please send your papers, if you have some, to the research topics that still we have time. And so we're going to be really happy to accept your work. So with that, I'm open to questions. Thanks.
Oh, thank you so much, Nico, for the excellent presentation of your research. I think we'll have the Q&A at the end. So.
Oh, okay, yeah.
Yeah. So to continue this very interesting conversation, I would now like to introduce our second speaker, Dr. Ho-Wan Jung. Dr. Ho is an assistant professor at the Department of Genetic Engineering at Dong-A University in Busan, South Korea. So thank you, Ho-Wan, and please feel free to share your screen, and the floor is yours.
Can you see my presentation?
Yes, we can see the screen.
Okay. Yep. Okay. So first of all, I really appreciate all staff and Dr. Nicolas Cecchini for giving me a chance to present my work. Also, thanks to all of you for attending this webinar. Today, I would like to introduce the role of the HEC1 and DODL and DDL in the plant immunity. Yep. Maybe the Nico give us the nice introduction. So I just briefly summarize my previous work. When I see the salicylic acid level in the distal lip tissue in the immunized plant, the salicylic acid level is not different from the mock immunized plant and from between mock immunized plant and the pathogen immunized plant. But the old explanation of the SAR in distal lip tissue, salicylic acid level is higher in the pathogen immunized plant than the mock immunized one. But in that case, I didn't see any increased salicylic acid before secondary pathogen infection. But after secondary pathogen infection, salicylic acid level is higher in the pathogen immunized plant, as like this. And thus, we conclude that the systemic acquired resistance SAR induction tightly correlates with the immune priming, as like the Nico said. Also, Nico showed this type of illustration, but as this illustration shows, successful immune priming depends on the strong and rapid expression of the immune response after secondary pathogen infection. When I studied in Dr. Jean Greenberg's lab at the University of Chicago, I also found this phenomenon in Arabidopsis. And I showed the last slide. How do plants regulate this immune priming is my fundamental question. To get simple answer to these questions, I have checked the acetylated histone H3 level in distal lip tissue after mock immunization or the SAR-inducing Pseudomonas immunization. As you can see here, the level of the acetylated histone H3 seems to be higher in pathogen immunized plant compared with the mock immunized plant. Also, in the promoter region of the ICS1 gene, the acetylated histone H3 protein were highly enriched in the pathogen immunized plant compared with the mock immunized one. I got this data about 15 years ago in Dr. Jean Greenberg's lab at the Chicago. Anyway, this data show the histone modification may be involved in systemic acquired resistance. And then I came back to Korea in 2010, and I'm continue in this research at Dongha University. As you already know, Drs. Konrad and Ton's group reported these two papers in Ember Reports and Plant Physiology. In these two papers, they demonstrate that the active histone marks increase at promoter region of working genes in systemic leaves of immunized plant. Also, Dr. Ton's group suggests that histone modification caused by SAR can be passed on to the next generation. However, at that period, we didn't know which genes are responsible for chromatin remodeling occurring in immune priming in plants. Like the title, today I introduce the role of the one-up histone acetyltransferases, the HEC1, acetyltransferase 1 in plant immunity. In plant, histone acetyltransferases were classified into 3 different groups. To identify the immune-related gene among this gene family, we collected and isolated tDNA insertion mutant during 4 years and screened the immune defective or enhanced immune mutant in Arabidopsis. As a result, we isolated HEC1 mutant as an enhanced immune mutant. When my team was studying the HEC1 gene at Dongha University, 2 papers were published in the Plant Cell and Nucleic Acid Research. In these 2 articles, they proposed that HEC1 play a positive role in plant immunity. However, my lab got totally different results from these 2 papers. I think that the environment condition is totally different in the Singapore and the Seoul National University in Seoul in Korea and Busan in Korea. So environment has different effects on the chromatin remodeling in plants and animals. Thus, it might be possible that the different biotic and abiotic conditions can cause differences among these independent experiments. When I started that experiment, my team initially used the Pseudomonas syringe strain, Pseudomonas syringe maculicola strain as a virulent pathogen. As you can see here, when bacterial titers were higher than 10 to the 6 CFU per lip disc, HEC1 mutant showed the resistance phenotype to virulent pathogen infection. However, other mutants, including HEC12, 4 different HEC mutants showed a wild-type likely susceptibility to virulent pathogen infection. Disease symptom was also reduced in this mutant. One interesting thing that we found is that the HEC1 often displayed the wild-type likely susceptibility when the bacterial titer in wild-type plant less than 10 to the 6 CFU per lip disc. This showed the possibility that environmental conditions are important for this type of pathophysiological study. That's why I guess that's why the 3 different groups propose a different function of the HEC1. Anyway, we conclude that only HEG1 among P300 CBP type histone acetyl transperases, only HEG1 participate in plant immunity. To confirm this resistant response of the HEG1, we use different mutant earlier and the different virulent strain. As you can see here, the HEG1 mutant were resistant to infection with these two different types of virulent Pseudomonas. But HEG1 mutant shows the wild-type-like resistance to avirulent pathogen infection carrying ABR-RP2 and carrying the ABR-RPS4. Also, my team checked the resistance to the ABR-RPM1 carrying resistance to Pseudomonas carrying the ABR-RPM1. At that time, the HEG1 also showed wild-type-like resistance. Thus, we believe that the HEG1 mutation conferred disease resistance on plants rather than the disease susceptibility. To see whether or not the enhanced resistance of the HEG1 was related to increased defense responses, we compared the gene expression and callus deposition in HEG1 mutant was those in the wild type. As you can see here, transcript level and PR1 and PR4 were dramatically higher in the HEG1 mutant compared to wild type after pathogen infection. Also, the FRK1 and NHL10 were strongly transcribed in the HEG1 mutant compared to wild type. Also, callus deposition is very dense compared to compared to wild type plant. However, the ICS1 and MPR1, the salicylic acid biosynthetic gene and salicylic acid responsiveness, and salicylic acid MPR1 is responsible for salicylic acid perception and responsiveness. But in these two genes, expression in the wild type in the mutant plant were comparable with those in the wild type. Thus, we conclude that the HEG1 mutant more sensitively respond to pathogen infection. Let's move on the system leaf of the HEG1. To examine the role of the HEG1 in SAR response, we infiltrated 10 mmol NGSO4 as a mock and also infiltrated ABR Pseudomonas strain carrying ABR-RT2 in the lower lip tissue prior to 2 days prior to challenge inoculation. We counted the number of bacteria on 3 days later, 3 days after secondary infection. Also, we determined several immune-related genes expression in these different time points. As you can see here, local immunization with the ABR Pseudomonas strain successfully induced ACR response in wild-type plant. However, local immunization of this ABR strain failed to enhance systemic resistance in these two HEG1 mutant early. This result shows that the mock immunization of local leaves was sufficient to express systemic resistance in HEG1. In other words, the HEG1 mutant might be in the prime state, as the Nicolas said, in case of the MOP, as like the MOM1. We also monitor the transcription of the immune-related gene in systemic leaves before secondary infection, then after secondary infection. When the wild type and mutant were immunized with avirulent pathogen here, we did not see any increased PR1, PR4, and NHL10 expression in HEG1 mutant compared to wild type plant. However, in case of the MOP immunization, After secondary infection with the virulent pathogen, PR1, PR4, and FRK1 and NHL10 genes expression was strongly expressed in HEG1 compared to wild-type plant. Once again, this data strongly suggests that the HEG1 mutant more sensitively responds to Pseudomonas infection. When my team has struggled with this result, different from those of other groups, we found a short perspective paper. In this paper, they proposed that these 5 different proteins may interact with the HEG1 in Arabidopsis. Using the tDNA mutant corresponding to this gene, we tested disease-resistant response. As you can see here, DDL and Dracula2 mutant only show the hypersusceptibility to virulent pathogen infection. Also, we confirm this susceptibility of the DDL mutant using these different mutant alleles. Thus, we conclude that the mutation of the DDL and Dracula2 caused the disease susceptibility to infection with the virulent Pseudomonas infection. What is the chat? Maybe it's wrong. No. I just wonder I'm wrong, but yeah. How can quit this chat window? I'm so sorry. Why I'm open this chat window?
Has it popped up on the screen or?
No.
Oh, okay.
Yeah, okay.
Okay.
I'm so sorry.
No worries.
The DDL is the one of the PROC-head associated domain protein. Also, the DDL binds to primary microRNA. And consequently, DDL participate in the microRNA biogenesis in Arabidopsis. Also, the PAR-related DDL is required for the plant immunity. Thus, we had an interest in the role of the DDL in HEG1 mutation-mediated plant immunity. To solve this question, first we crossed the HEG1 mutant with the DDL mutant, and then monitored the growth and developmental phenotype and then disease resistance to virulent pathogen infection. It is already known that both the HEG1 and DDL mutant show the late flowering phenotype. This late flowering phenotype became more severe in the HEG1 and DDL double mutant. We grew this mutant plant under neutral and short day condition. We got the similar results in the both cases. As Professors Ribochan and the Finnish group published earlier, the DDL mutant was susceptible to virulent pathogen infection. Interestingly, the HEG1 DDL mutant showed a wild-type-like resistance, unlike their parents. Thus, we conclude that the DDL regulates plant immune response in the opposite way to HEG1. But this result also came up as the next hurdle my team will need to clear. How do HEG1 and DDL control the immune priming in plant? We assume the different possibility. Scenario 1 is that HEG1 may help the transcription of the MIR gene targeted to immune-related genes. Scenario 2 and scenario 3 and scenario 4 is that the HEC1 may trigger transcription of negative regulator, and the PAR-related DOTIL is involved in basal defense response, but that basal defense response might be independent from the HEC1. First of all, we are trying to examine the scenario 1 to check if the HEC1 can also control the microRNA accumulation in Arabidopsis. We performed the microRNA-seq analysis in non-infected wild type and HEC1 single mutant and HEC1 and DDL double mutant with the pathogen infection. This table shows the brief summary of our microRNA analysis. By this analysis, we found about 20 differentially expressed microRNA. Here is the list of the differentially expressed microRNA, and the microRNA are tightly associated with the immune responses highlighted in yellow. Now we are checking the expression pattern of this microRNA in wild type and mutant with and without the pathogen infection to test our first scenario. Today, I cannot show the conclusive result about the role of the HEC1 and DOTIL in plant immunity because I'm still working in this period. Also, some different group published the opposite result. But because of that opposite result, my team is interpreting these results very carefully. So that's why today's my talk is not complete. Anyway, the main take-home message is the HEC1 is negatively regulates partial defense responses in Arabidopsis, unlike the previous report from the other groups. Also, the HEC1 mutant seems to be the prime state. Considering the molecular and functional interaction between the HEC1 and DOTIL mutant, DOTIL implant, these two genes seem to have the additive effect on the plant growth and development because the double mutant shows the late flowering phenotype. But in the point of the plant immunity, HEC1 may be involved in suppression of the immune responses to keep the proper energy allocation. On the other hand, the DOTIL might be regulate the immune responses. Through testing every possibility I showed earlier, my team is going to reveal the role of the HEC1 and DOTIL in plant immunity in the near future. Thanks to all the members in my lab, the plant immunity and epigenetics, and especially I thanks to Professor Cecil Sogonjang and Dr. Wan-Hee Kim in Seoul National University. Thanks to all of you. Thank you for your attention. Thank you.
Okay, thank you so much, Ho-Wan. So I'd like to thank both Nico and Ho-Wan for their very interesting talk on what is happening at the molecular level. And I'm sure that this current research will further develop the field. So I would like to now open the floor for some questions. If anyone has any questions, feel free to unmute yourself and to ask Nico or Ho-Wan something.
Hi, Nico.
Hi, everyone.
Yeah, Nico, thank you for your talk. And TSI gene is just a marker to check the histone demethylation.
Well, in this talk, yes, but actually TSI is a transposon. So yeah, the mechanism is like is transposon in the pericentromer region. They are overexpressed in the mutant, and also the same happens during infection. I didn't show that. It was published before from another member of the group. And actually what happens, the smaller, there are several ideas, but then one of the ideas is that these transposons are expressed, so they start to trigger the RDBM remethylation of the region. So because you want to turn off these guys there. So all these small RNAs that are generated, they also have perfect match with immune receptors promoters, actually. So the idea is that all the machinery for RDBM is used to silence the centromeric transcription that happens. So then you don't have small RNAs or the RDBM machinery to repress the immune receptors. So then the immune receptors goes up. That's one idea. It could be also that you have long non-coding RNAs expressed from the centromeres, and not actually like recently it was published that it could be that some small RNAs, like the 21 nucleotide type, they can go with AG1 and actually prime the promoters of the immune receptor. So it could be any of those, the idea. So it's not just a marker. I mean, TSI and another transposon, they are doing something in they are transactivating the immune receptors.
Okay. And can I ask one more?
Yeah, sure.
Is minimum expressed plant? Does minimum expressed plant shows any developmental defects or?
No, actually, I didn't show that because we didn't quantify, but the most amazing thing, it's not that amazing. It looks that in some environmental conditions, the plants looks bigger. That it makes sense because it's like the threshold between defense or growth, no? So because it's like a plant that it looks like it never acquired priming, not even in a natural condition when the plants grow in soil and just aging, that you have like an increase in defense. It may be possible that actually minimum it doesn't age, you know, in the terms of defense. That's what I have in my mind actually. That's why it's bigger, but only in some conditions. Not is exactly the same as water.
Yep. Thank you.
Okay. Any other questions? Anyone from the audience? Feel free to type questions in the chat box if you wish.
Sorry, I have one question for Nico.
Yeah.
First of all, thank you for the talk. It was super interesting. Just correct me if I'm wrong, but I was amazed by the phenotype in terms of the molecular part of the minimum of the truncated version of MOM1.
Yes.
I was wondering, have you done any research on what is going on there? I mean, it seems that it's completely unregulated. So have you checked what is going on all those sequences that are missing in this truncated part, and what are their role in regulating MOM1?
Okay, we're going to into the response. The first thing that we are doing right now is actually to check the transcription of this transgene, because I didn't say it, but the transcript has the native promoter. So we want to check actually if the transcription is changing as MOM1 or not, because it's a transgene, so maybe actually it doesn't go down like MOM. That could be why it's happening that. But of course it doesn't have all these domains, but nobody knows exactly what they do. Some of them is like a platform to interacting with another factors that are implicated in chromatin remodeling, like the MORCs proteins that also are implicated in defense. So it easily can change it doesn't interact anymore. So the only thing that it does is just repress. That's one idea. The other idea, it could be that it's so small, the protein, that just goes inside the nucleus more, compared with MOM1. That could be another answer. But I don't know actually what all the other domains. What I can tell you is that it has all these domains that are highly similar to other chromatin remodeling factors, like the I don't know, the family of these remodelers, but actually it doesn't have the catalytic function of those because it doesn't have the right amino acid. So it's just the domains, but it's not like the ATPase activity, for example. It doesn't have it. So it's doing something else, maybe like generating the complex that require for that. So anyway, what we know is that actually it's completely unregulated protein, and it doesn't function like the native protein. Just it's like a constitutive negative regulator of the pericentromeric regions. I don't know if I answered.
No. Yeah, I absolutely understand that this is ultra difficult to do and requires a lot of research. I was just curious. Thank you very much.
No, thank you.
Okay, and Ho-Wan, I do have a question for you. I was just wondering what kinds of genes would be targets of HAC1 during the immunity?
Unfortunately, I didn't get any concrete result yet, but I think some microRNA HEG1 might be regulated, some microRNA with DOD during the pathogen infection or the before pathogen infection to suppress the improper activation of the immune responses.
Okay, thank you.
Yep.
Right. All right, so okay, so I don't know if anyone else has any additional questions. We are sort of running a little bit over time, but yeah, so I would just like to say that we have our contact page here. We've added our email address. So if anyone has any other additional questions for Nico or Ho-Wan, please, feel free to email us and we will add the questions to the comments section of our YouTube video. So yeah, and please feel free to reach out to our journal if you're interested with working with us or collaborating with us. Please follow us on Twitter and visit our webpage for the latest updates. If you would like to do an article collection with us, we are very happy to hear from you. And as Darina mentioned earlier, the 2022 Global Plant Council Prize is also open. So do get in touch with us for more information if you're interested. And if no one has any other questions, then I think it's time to wrap up the webinar. So before we say goodbye, I would again like to thank our speakers, Isabel, Nico, Ho-Wan. Thank you again so much for the lovely introduction to the GPC and also the great talk and the discussion. Thank you for agreeing to do this webinar with our journal. It was really great. I hope everyone had fun and learned a lot. I know I definitely did. And our team, and also I hope all the attendees had a good time. So yeah, so thank you for your participation, and I hope you have a great afternoon and a great rest of the week. Yeah, so thank you.